Guys, how are you? Well, today we're going to talk about two subjects. One is graph theory and the other one is systems biology. You're going to understand later why we have to explain a little bit of graph theory before we talk about systems biology, which is a very important field in modern fields in life science. Okay, uh, first, why should we care about graphs? And I know that you are asking, like, first, what the hell is a graph? Well, graphs are a set of edges and nodes. Nodes can also be called vertices, and these are the edges, the connections, the link between two nodes, okay? So a network can be represented by a graph. Okay, so now the question is, why should we care about networks? Well, one important reason is that all life forms are connected into a huge network, okay? Unless synthetic biology already advanced a lot, all the life forms are connected. So if you see like the tree of life, you can see that all organisms are there connected into a huge network. And if you take a zoom here, you can see, for example, the three organisms that share some characteristics. Okay, for example, these two here are mammals, and these three here are chordata. Okay, they have similar characteristics that put them into a similar group. And when you start like organize the living beings uh, based on characteristics, you can actually go back and construct a phylogenetic tree that group organisms that have similar characteristics. And these characteristics are actually the phenotype that you learned already. But what is underneath the phenotype is the genotype, the DNA sequence that make up a cell and therefore an organism or individual. Okay, so if we take the phenotype, which are the characteristics, we can also think that there was a genotype that explains this phenotype. And of course, if we could sequence during the millions and millions, actually billions of years of evolution, we could see the mutations happening, explaining in the ancestors why we have the species that we have today. Okay, but of course, you learned that it's not that simple. It's not that there is a simple arrow coming from genotype to phenotype because there are epigenetics regulations, no coding RNA, etc. And not only life, but almost everything else is connected. For example, air, flights, social medias, and gene networks. Okay, and let me explain a little bit about graph theory, so you're gonna understand why we need this to understand how the life is organized. So graph theory starts with this guy Leonhard Euler that live in Königsberg in Prussia. In that small city, they like to play a game uh, because they have in the city seven bridges. So the problem was to find a walk through the city that would cross each bridge once and only once. Okay, so I'm gonna draw here the seven bridges, and here are the landmasses that are connecting. The game was to try to find a path where you walk to all the bridge, but only walking once. So you will see that if you go here, you can go here, and then here, 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 and then it finished because you're gonna have to pass this bridge again if you want to go here. Okay, so you realize quickly that there is no solution. And Leonhard Euler was able to see this problem as a graph and actually put in a mathematical formula that explains why it's impossible to find a simple path that walks through all this bridge. Okay, that was known later as the Eulerian path that was never solved until the mayor built this another bridge, and now it's easy to do this path. Okay, so when you have like without this bridge, you can see, for example, if the node has an odd or even number of bridge. So here is 1, 2, 3. So this node here has an odd number of bridges. This one has 5 bridges, odd number. This one has also 3, odd number, and this one has also 3, odd number. So Leonardo Euler showed that actually you to find this path, you have to have at least a maximum two nodes with odd numbers. So when the mayor builds this bridge here, now you have this node here with 4 bridge, which is an even number. This also has a 4 bridge, which is an even number. Now you can see that it's possible to find the Eulerian path. But look, you cannot start from anywhere. For example, if you start from these nodes here, you go this here, and then here, 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 and here. You see that you cannot connect here. So you have to start with the nodes that have odd number of bridge. So if you start here, you can go down, up, down, up, down, up, down, and then left. You see, you always end up from one node with odd number to another node with odd number. So that's the Eulerian path. Of course, that was not the only thing that Euler discovered. He's also known for his work in mechanics, fluid dynamics, optics, astronomy, music theory, and that's why even Google celebrates his anniversary. Well, let me explain some technical terms before we advance. So these are this is a directed graph and this is an undirected graph. You see the difference that here you have a direction, whereas here you don't have any direction. And why this is relevant? For example, imagine that you have a kinase that phosphorylates this protein here. So you see that this kinase phosphorylates this protein, but this protein does not phosphorylate this kinase. Whereas this case here could be two kinases that one can phosphorylate the other. Here, you don't have this direction information. So for example, one example for this could be two proteins that physically interact into a complex. Okay, so it doesn't matter if one is connecting to two or two is connecting to one. The only thing that matters is that one and two connect to each other to make a protein complex that will do something in the cell. Okay, and that's important because when you do mathematical models using graphs, you have to know if you're dealing with a direct graph or undirected graph. What about the degree? The degree of a vertex is the number of edges that incident on it. So for example, the degree of the vertex 2 is actually 3 because you have 1, 2, 3 connections, 3 edges, okay? And the degree of the node 3 is 1, you only have 1. The degree of 4 is 2, you have 2 connections here. A walk is a sequence of nodes. So if you go here to here, it's a walk. If you go here, here, and here, and here, and here, it's a walk. And a simple path is a walk with no repeated nodes. This is the Eulerian path. So you can go from this node to this one, this one, this one, this one. That's it. But you cannot go this one, this one, this one, and go back or go making a circle, because that's not going to be a simple path because you are repeating the nodes. Okay, and again, this is important if you try to understand the pathway, signaling pathways. Okay, 200 years later in Hungary, these guys, Paul Erdős, as you can see, he's a very normal guy. He had his doctorate in mathematics at the age of 21, 1,500 papers, and he was so famous that there was this thing called Erdős number. Okay, that's the number of people that you have to publish to be close to publish with Erdős, the greatest mathematician. So for example, if I publish a paper with him, my Erdős number is 1. If I publish with someone that published with Erdős, my Erdős number is 2. So it was a matter of prestige to say that, oh, my Erdős number is 3, whereas your Erdős number is 7. So that means you have 7 people to be connected to Erdős. So Alfred Rényi, he had 32 papers with Erdős, so his Erdős number is 1, and 8 of them were about how networks are formed. So that became the models for generating a random graph. And how can we generate a random graph? Imagine this situation like this. I'm pretty sure you guys already been in a situation like that. It's very awkward. So you have to, usually you talk to 2 strangers, 3 strangers, right? You don't talk to everyone in the cocktail party. And what happens that imagine that a waiter comes to you and say like, look, the best wine in the party is the one with the green bottle. Don't tell everyone. But then you only tell the two people that you are talking to. And these two people will talk to another two people and so forth, so on. How long do you think it's gonna last, this green bottle wine? It's gonna go very quickly, right? Because you're gonna see that although each person only talked to a few other people, at the end, everyone will know about this green bottle. That's the power of gossip, okay? And how do you prove this? How do you create experimentally a random graph? Look how elegant they did. So what they did was to create the nodes, these circles here, and then for each pair of nodes, they roll the dice. If the dice rolls a 6, they make a connections between the two nodes and then move to another pair of nodes. And if the dice rolls any other number, they do not make any connection and move to the next pair of nodes. So imagine that they were rolling the dice thousands of times to try to see if they create a random graph. And when they do this, they see that although the frequency, the probability of you making a connection, which is 1 in 6, because only if you roll 6 out of 6 possibilities, you make a connection. So although you have a degree which is close to 2, you see most of the nodes have a degree of 2. At the end, everything is connected. So all the nodes somehow is connected to each other. So Erdős assumes that God must like to play dice. And then Albert Einstein, which has an Erdős numbers of 2, was convinced of the opposite. So God does not play dice with the universe. Probably you saw this sentence in a completely different context. Well, we create this experiments for you using a computer. So Lucas, my students did create this network generator tool where you can find in my website. I'm gonna put in the comments of the YouTube so you can play with this tool. What you're gonna see that you can create a random graph, or you can create later a graph with preferential attachment. But when you create a random graph, you can see, for example, if there is any isolated nodes, you're going to see that it's very rare. And also, what is the number of connections per node that you see? Is roughly the same, or they are very different to each other? And then I want you guys to think about examples of random graphs. Well, one example could be the highway system in US. So as you can see, most of the cities will have around 2 highways, or some of them you're gonna have a little bit more. But doesn't matter the size of the city, doesn't matter if it's New York or Madison, you're not gonna see a city that has 35 highways and most of the city has 0 highways. At least they're gonna have similar number of highways going in or going out. Another example could be the brain. So you see that in your brain you have 100,000 neurons connected to one neuron, and this one neuron connects to another 100,000 neurons. Okay, imagine now 100 billion neurons in your brain doing this network. Another one is the zombie apocalypse. So you don't see in the movies one zombie infecting 200 people and 99 zombies doing nothing. What you see is that one zombie infects 2 people, this and then these 2 people will infect two other people, each one. So in that way, the whole world is gonna be infected. So the main properties of the random graph is that all nodes will have approximately the same degree, the same number of connections, and they will follow Poisson distribution. So this is a normal distribution that you know that's done with continuous variable, and this is the Poisson distribution, which is uh, using discrete variables, age in years, or in this case, the number of connections. Okay, here you see that the density, most of the nodes will have between 4 to 6 connections. And you're going to see very few nodes that have 0 connection and very few nodes that have 10 connections. So if that was, of course, a distribution of degree in a network. But random networks will follow this distribution here. Another important concept is the degree of separation. You can calculate, for example, what is the minimum path between two nodes. So, for example, the degree of separation of A and B is 1. The degree of separation of A and C is 2, because you can go to C, going to B, and here. So 1, 2, instead of 1, 2, 3, 4. Okay, so because the shortest path is 2, use the degree of separation of AC is 2 and not 4. And when you do this for all combinations of nodes, you can calculate the mean degree of separation of the whole network. And you see why this is relevant. For example, what is the degree of separation of everyone in Facebook? So, in other words, how many people you have to be connected with the Pope or with Brad Pitt? You're going to see that it's going to be less than 6. So meaning that you know someone that knows someone that knows someone that knows the Pope. Okay, so in average we are away from each other to 6 people, less than 6. What about authors in PubMed? You're going to see that is around 5. So that means that having an Erdős number of 5 does not mean anything, because that's the average of authorship network. And what about the pages in the internet? In the year 2000, where you only have 1 billion pages, it was around 19. And what about the degree of separation of all the genes? Well, look, it's only 3. So that means that you can connect any gene with any other gene through 3 connections. So if you are analyzing a data set on Alzheimer, and the gene that you found has nothing to do with Alzheimer, what people usually do is say, like, okay, my gene has nothing to do with Alzheimer, but my genes connect to this gene that someone described in 1976, and this gene was connected to another gene that someone described in 1984, and then this gene is connected to another gene that someone described in the year 2000, and that gene was related to Alzheimer. Therefore, my gene is related to Alzheimer because my gene is connected to A, which is connected to B, which is connected to C, and C is connected to Alzheimer. You see that there's unfair to make this analysis. You cannot interpret your results based on this, because on average, all the genes are connected to each other through 3 connections. So that means that you can connect any biological process with any gene in a very few number of steps. So that's why systems biology is important, because you can only make sure that your findings is relevant if you look at the whole network. You can also check, for example, in the network generator, what is the average link per node of your network that you create. What is the average shortest path between 2 nodes, which is the degree of separation, and what is the biggest distance between any given pair, and what are they? Okay, you're going to see on the right that you have these statistics shown for you, or on the left. And then you can ask yourself, you can play with the tool, and you can increase the number of nodes, you can increase the connections by increasing the probability of you making the connections. And then you can ask yourself, what caused the bigger impact on the degree of separation, the number of nodes or the number of links per nodes? Okay, imagine what is the degree of separation when you create, for example, a network with 10 nodes and two connections per node. And then what is the degree of separation when you put 30 nodes, 3 times more nodes, but the same number of connections per node, 2 connections per node? And now you compare with, if you have 30 nodes and 2 connections per node, and 30 nodes, the same number of nodes, but 4 connections per node. So you increase the number of links per node. And then you see what is the change in the degree of separation, what increases the most the degree of separation. And that's important, because Random graphs are more democratic. So most of the nodes will have similar number of links. That's the Poisson distribution that I showed. But does this represent most of the real networks? Is the internet a random graph? So when you create a blog about French cuisine, do you think your blog is going to have the same number of links pointing to your blog as the Amazon or Google? No, right? What about societies? Do you think that your 1,000 friends in Facebook is going to be close to the number of followers of Anita, for example? Or the gene networks? Do you think that all the genes are connected the same way? And then you see that no, actually, most of the networks have these hubs, which are the nodes that are very, very, very connected, much more than any other nodes in your network. So examples of nodes are p53, the gene p53, the protein, the Google, Lula, etc. And then you're gonna understand that. So one of the people that's realized that we don't live in a democratic network is Vilfredo Pareto. He was an Italian economist that liked to do some gardening. So he realized that 80% of the peas came from 20% of the pea pods. 80% of Italy's lands were in the hands of the 20% of the Italians. And that became the Pareto principle, or the 80-20 rule. And that it's like the Murphy Law. You see that 80% of the crimes are committed by 20% of the criminals. 80% of companies' profits come from 20% of its customers. 80% of links point to 15% of websites, and so forth, so on. Okay, so that shows that actually it's a small fraction of nodes that explain most of the stuff. So if you calculate the distribution of the links in a network like this, you're gonna see that they don't have a Poisson distribution, they have a power law distribution. So for example, this is a random graph where you have the number of cities, for example, and the number of the cities are the nodes and the highways are the edges, the connections. So you see that most of the cities will have similar number of highways, like 2 to 4. You're gonna see almost no city with no highway and no city with 100 highways. Okay, but if you look at the same map, but now using the air flights connections, you're gonna see that you find some hubs: Chicago, Houston, Atlanta, New York, because these are the headquarters of airline companies. So these cities are the hubs. Most of the cities will have a low number of flights going in or going out, but a very few number of cities will have a huge amount of flights going in or out. These are the hubs. Okay, so hubs, imagine that you are walking on Paulista Avenue and then you see someone with 300 meters high. That's how different they are from you. So if you look at a biological network, you're going to see, for example, that C. elegans or Drosophila, if you connect all these biological components, they follow a power law distribution. And how these networks are formed. So they usually begin with a random network that already exists, and then they grow. So one characteristic is that the network grows, and they do not grow normally. They do not grow at random. The new nodes that's going to connect to this random graph, they are going to preferentially connect to nodes with higher number of links. Okay, so if a new node is coming to your network, it's going to choose the nodes that already have a lot of connections. So that means that probably the nodes that already have a lot of links, they're going to have more links, right? But these are not the only two properties of real networks. Let me explain why. So let's take the email user accounts. So in the 2005, there were many more users of Hotmail than Gmail email accounts. Okay, so if your aunt decides to create email accounts, do you agree that in 2010, if she decided that, she would choose Hotmail because Hotmail was already the service with the more number of users? So you would expect that if the only two properties of these networks were growth and preferential link, Hotmails will be users going to be higher. So the reach always become richer. But that's not what happens. The Gmail users increase and the Hotmail users decrease. And that's because the fitness of the nodes is also essential in attracts new nodes. Okay, it's not only these two, but also the fitness. You can all and then you're gonna realize that not always the rich become richer in this type of network. This network is called scale-free networks because you cannot think in a scale to measure this network because of the hubs. So you can include, for example, the fitness in the formula in this model. So this model without the fitness, the only thing that matter is the probability that you're going to make a connection depends on the number of connections of the nodes that you are about to connect already has divided by the total number of links in the neighbor nodes. But if you put the fitness here, you're going to put a weight on the nodes. So a new node will have a higher probability of connecting to a node that have low connections, but has a high fitness. How can you apply this theory to DNA sequencing? So remember the problem that when you sequence the DNA, you actually produce a lot of reads, which are fragments of the DNA that you have to assemble into a genome. And the problem is how do you do this overlap between several millions of reads? So most of the modern assemblers use the De Bruijn graph approach. So they use the graph theory to do this. And the way I'm gonna exemplify to you is using MPB Lyric. Okay, because if I put here ATCG, it's gonna be even harder. So imagine that this is the genome that you want to sequence. I'm just gonna remove the space, and that's gonna be the genome that you want to sequence. So remember that when you sequence the genome, you actually generate some millions of random small reads. And this is the invisible part that you want to sequence. And what you have to do is to assemble all these reads into the original sequence. The problem is that if you compare these reads with all the other reads, and then these reads with all the other reads, and these reads with all the other reads, 10 million times, you're going to have a number of compares that is overwhelming. Not even computers can run this analysis. And the solution is representing this data into graphs. And how the assembler do this? So the step one is to convert the reads into sequence of size k. So this is the k-mers. So for example, if you split each read in k-mers of 3, you're going to have one node here with these 3 letters, the other nodes here, the other nodes here, and the other nodes here. So you make a graph using only this, a directed graph. And then same thing here, and you do this for all 10 million reads. And then you can use these nodes to index them. So it's much easier to find if you create an index than if you try to find if you index the whole stuff. It's quicker. When you do this, later you have to simplify the graph as much as possible. You can combine repetitive regions, which are bigger than the k-mers, and find these kinds of things here. You can then remove the redundancy and create the consensus. And that's going to be what you're going to get in the final assembly using k-mers of 3. And then you repeat the same process with k-mers of 4, k-mers of 5. And for example, if you use a k-mers of 20, you're going to have this assembly here, which is good. Say like, oh, why we didn't use the highest k-mers possible? First, because it's going to be more computational intensive if you have longer k-mers, because then the index is going to be too big. Second is that there are some sequencing errors that are gonna be more frequent if you have a longer k-meres. So for example, if you have a sequencing error here, instead of a T, you have a V. If you have a k-meres of 3, only 3 of the nodes are gonna have these sequencing errors, and most will not have. Whereas if you have a k-meres of 9, 100% of them are gonna have the sequencing error, okay? And that's how you can use graph theory to sequence the genome. And actually, you can play with networks using the tool that I showed you in the previous class, which is called Network Analyst. And you can also visualize the networks using GAFI, which is a free software that can make beautiful, beautiful networks like this. You only have to learn how to play a little bit. And that's when we finally start talking about the biology here, which is a systems biology. And you already know that we characterize already the the biological components. We know that we have a 3.2 billion base pairs genome. We have more than 150,000 transcripts, more than 1 million proteins, etc. The problem now is how can we characterize their role and how to elucidate how they are organized. And the way we do this is using network. And that's why graph theory is important. So if you take a look in the East transcription factor binding network, so where the nodes are transcription factors and the genes that are transcribed by them, the protein-protein interaction network, the phosphorylation network, metabolic network, genetic interactions. You see that inside the cells, we actually have huge and complex networks. And the way we scientists were trained was trained using the reductionism approach. Reductionism is based on the idea that complex systems or biological phenomena can be explained by analyzing its simplest components. Okay, so imagine that if you so Francis Crick used to say in 1966 that the ultimate aim of the modern movement in biology is to explain all biology in terms of physics and chemistry. So if I want to understand how human beings works, I have to break this into tissues and then break this into cells and then break the cells into molecules and then break the molecules into atoms and then break the atoms into physics and chemistry, okay? And you can even break this even further, and that's going to become mathematics and then philosophy, okay? So basically they thought that if you want to understand the complex, you have to reduce the most and understand the single components. That's the reductionist methods. And what became clear is actually the proper method is the holistic method, which is based on the idea that biological systems and their properties should be viewed as a whole, not as a collection of parts. So the whole is greater than the sum of its parts. So you can break down the whole airplane. You can have all the screws, all the pieces together, and you're not gonna see how that machine can fly if you only analyze the screw or the metal planes. So imagine you have a lot of blindfolded scientists analyzing an elephant. Depending on where the scientist is touching, it's gonna conclude a completely different stuff. For example, it's gonna say, "Oh, it's a rope, it's a wall, it's a fan, it's a spear, it's a snake, it's a tree." Okay, because no one is seeing the whole. But what happens that high throughput technology now allows us to see the whole. The problem is how to analyze this huge amount of information. So there are two main approaches of systems biology. One is called top-down, where you start from the most complex one and then you reduce the information to try to understand what's going on. And the other one is bottom-up. So you start with the parts and then you integrate them to try to understand the whole. Okay? So this is the bottom-up systems biology, where you have, for example, a reaction here, then you have another reaction there and another one there, and then you integrate all the reactions and try to understand the organism. Okay, and that became like a hypothesis that's okay, what if I block this reaction, what's going to happen? And then you use your model to try to validate if your hypothesis is true. Okay, so that's the bottom up. And biochemistry was created in this way. So you first characterize the parts, the reactions, and then you try to integrate it into a metabolic pathway. The top-down systems biology is the one that I do, which is you start from the complex, the organism, the tissue, or the cell, and then you measure the whole stuff using omics data, and then you use mathematical models, statistical models, to try to reduce this to small things that you can create hypotheses, perturb the system, and see if this is actually happening. What are the properties of systems biology? Well, we can think in three properties: emergency, robustness, and modularity. Emergency is the example that I already gave to you, like how many protein coding genes exist in the human genome. And then you're going to see that we have less protein coding genes than the rice, okay? So this is the G-value paradox. And the reason is that the biological complexity is associated with how the genes interact with each other rather than the number of genes that you have. So another way of exemplify emergency is if you think in a neuron that can connect to another neuron through a synapse, and that's going to be part of a nerve, that's going to be part of a brain. So when you connect a lot of simple components, and you use millions and millions of interactions, that can emerge a complex things such as the thoughts, okay? So complex systems and patterns can arise from multiple simple interactions. So this is the emergency that happens when you have systems biology. The other one is robustness. Robustness, so for example, you can exemplify here. Imagine that you have a simple pathway like this. You have the growth factors that bind to the receptors that triggers this signaling cascades and that's have an effect. If you use a drug that blocks this protein here, you will see that you're going to block the whole stuff, right? Everything that is downstream is going to be blocked. If you use a drug that blocks only these proteins here, you're going to stop this signaling, but you're not going to stop this one. Okay, so that means that this pathway is not robust. It's very easy to stop everything. But the thing is that because we have a very complex network, the system is very robust. It's very hard for you to think that you're going to block one protein is going to have a large effect here. Why? Because biological systems are relatively stable against perturbations, stochastic events, or genetic variations. Feedbacks due to feedbacks, checkpoints, controls, and redundancy. And the last point is the modularity. The same way the internet pages are connected to each other into modules. So here are the people that like Game of Thrones. Here are the people that like Frozen. Here are the people that like Black Mirror. Okay, so same thing happens with the molecular networks. So if you look at the genes that are related to ribosome biogenesis or assemble, they form a module, a highly connected group of nodes. Here's another module related to cytokinesis. And you understand that if I remove these nodes here, the cytokinase probably is not going to be affected. So that's also has to do with robustness. But if you remove these proteins here that are actually connecting different modules, then you can affect the biological systems. And that's why systems biology is important, because you will know where you can attack or try to inhibit or modulate. So as a summary, systems biology is an interdisciplinary field because it utilizes mathematics, computational science, molecular biology. It monitors all genes, proteins, and information pathway responses. It integrates many multi-scale types of biological information. So you can think not only in genes, but also proteins, epigenetics markers, metabolites, and it uses a more holistic perspective approach to biological research. And it also requires mathematical models that describe the structures of the system and its response to individual perturbation. So systems biology is actually what we do in the lab, and it's a very important concepts in area to understand. Okay, so even if you go to industry or stay in academia, understands that systems biology is the modern way of looking at biological systems is completely relevant. Okay, hope you liked. See you next class.